Dr. Patrick Fahey from the Queensland Herbarium. He's a restoration and conservation geneticist, has worked at the University of Melbourne and also worked in the Research Centre for Ecosystem Resilience at Botanic Gardens of Sydney. Patrick's going to talk on conservation genomics of Eucalyptus camaldulensis and the hunter catchment in support of genetically informed seed sourcing. Thanks, Patrick.
Right, let's just get this working. Can't really see it. That's a good start. All right. Yes, good afternoon, everybody. I'm taking off my current hat working at the Queensland Herbarium and putting on my old one, where I was down in Sydney working with Risa, to present some work we did in the Hunter Valley on Eucalyptus camaldulensis. So a bit of background on the project. I'm sure most of you, unless you're from Tasmania, are familiar with Eucalyptus camaldulensis. It's the most widespread eucalyptus species in Australia and is an iconic part of many landscapes. So riparian species lines the Murray-Darling Basin, all those large trees you see down there. So despite being very widespread, particular populations have been quite negatively impacted since European arrival, one of which is the population in the Hunter River catchment in New South Wales. So this is an interesting population because it's the only catchment in New South Wales which flows into the Pacific Ocean where you get the species. The rest of it's inland, Murray-Darling Basin basically. But also being in the Hunter Valley, experienced very heavy land clearing, land use change, mainly in this case due to agriculture. And so currently there's only 1,000 mature individuals remaining in the catchment, which has it listed as a threatened population under the state legislation. Out of this, being an iconic species, there's a lot of community interest, a lot of interest from Saving Our Species program, that sort of thing. So from that interest, it sort of became known that there was sort of a need for a genetic study of this population to address a few key uncertainties around it. First of which is there was uncertainty exactly which subspecies was growing in the catchment. There's many subspecies of camaldulensis across Australia, and there have been some mumbles about what was in the hunter. There's also, very small population that was greatly reduced from what it once was, which leads to a bit of uncertainty around how much genetic diversity is actually remaining. Have we only got very little of what was once there left? And how does that affect how it should be managed? And then, there's, as I said, a lot of community interest here in restoring it, replanting it into habitat. And, genetic knowledge can help ensure that those activities are as successful as possible. As Mauricio outlined, they achieve self-sustaining populations that, reproduce and, do their own thing into, millions of years into the future. And so that's particularly important for a eucalypt as well, given their breeding system is quite strongly outcrossing. So that led to a project with 6 main aims, which basically correspond to those questions loosely. The first is to investigate the extent of hybridization between Eucalyptus camaldulensis and other red gum species, both across New South Wales and in the Hunter, because it's known you see hybrids in the field quite often, and that might play into how you source seed, because you don't want to be sourcing that hybrid seed, planting that out, thinking you're planting a river red gum population when you're not really. Also, as I said, determine what subspecies is present in the Hunter Valley to ensure we're restoring the right subspecies. Assess the genetic distinctiveness of the Hunter Valley. As I said, it's the only place sort of on eastern New South Wales where you get the species. This question, is it genetically different? Is it responding to climate different? That sort of thing. Want to assess the genetic health of the remnant stands, and because that can affect seed sourcing. As I'll get to later, you don't want to go source seed from a lone eucalypt tree. You can get really bad seed that's going to perform really poorly. And then local landcare group, Bulk Broga Landcare Group, had collected seed from I think 10 trees along Wallumbye Brook. They'd maintained their seed collections as 10 independent maternal lines, so we were able to get samples of those from the nursery and test the genetic diversity and representativeness of their seed collections. And finally, as Sophia was outlining, you use all that information to sort of develop optimal seed sourcing strategies to empower restoration that leads to these healthy, self-sustaining populations. So the rest of the talk here is basically just going through the outcomes of the project and how that can inform on-ground activities. So aim 1, looking at hybridization between red gums across New South Wales. So you probably can't read that, it's a bit small, but basically Eucalyptus camaldulensis is in blue, the river red gum. We've got other red gum species we've got sampled here. I guess I should start with a bit of background on the genetic data. Won't go into it too much, but each point there is an individual from the wild with genotype using DartSeq, like Sophia outlined. So, you know, we've got nearly, I think we actually have over 1,000 individuals in there from across New South Wales. Coloration on the map matches what's in the plot. I'm not going to look too much at the map, it's not relevant to this talk, but basically in blue we've got Eucalyptus camaldulensis, then in dark green we have hybrids between camaldulensis and blakeleyi. You get them quite often in the Murray-Darling Basin, that's widely recorded. In the, what colour is that? The dark brown black, you have hybrids with Tereticornis. Those two species do overlap in a few places. We picked up a few hybrids there. And perhaps most importantly for the Hunter Valley question is these ones in teal, which are hybrids between camaldulensis and amplifolia, which only overlap in distribution in the Hunter Valley. So it's the only place you can get those hybrids, and it turns out all of those hybrids are along Wollombai Brook, where the local land care group was working. So this is just reiterating similar data. So this is a structure plot. So this is a similar analysis to the Leah Sophia showed, but basically each bar here is a single individual. And then it's proportionally assigned to different genetic clusters based on how many of the alleles it has fit into that genetic cluster, basically. So it's all coloured consistently. So orange is amplifolia and the genetic cluster that corresponds to that. So you can see here over at the far, well, not quite the far left, but the far left for the camaldulensis sites, we have elevated Eucalyptus amplifolia genetics in those populations. We actually have a few F1 hybrids where one parent was amplifolia, the other was camaldulensis. But the other thing we're seeing is all these sites along Wollombai Brook where the land care group is working have historic introgression. So probably when the red gum was colonising Wollombai Brook, there was an ancient hybridisation event, and those alleles have persisted in that population to the present day. Also importantly, the far right sites for Eucalyptus camaldulensis from Apple Tree Flat and Bylong, they're the sites where there was questions over subspecies assignment. Turns out they are morphologically different, but that's not because they're different subspecies, that's because they're hybrids with blakeleyi tereticornis. Can't tell those two species apart in the Hunter, but yeah. All right, so that's good. Move on to the subspecies in New South Wales. So here I'm just showing a map of all our genotype sites for Eucalyptus camaldulensis only, coloured by subspecies assignment by genetic data. If you're familiar with the species, you know that genetic assignment matches the known distribution of the different subspecies. So right through the Murray-Darling Basin we get Eucalyptus camaldulensis, subspecies camaldulensis, and everything in the Hunter belongs to that subspecies. So great, we've answered that question. We know there's only one subspecies in the Hunter, and it's the common Murray-Darling one. We also have Arida, which occurs along ephemeral waterways in the far northwest of the state, but is very common through inland Australia. And we have supposed acuter stands in the northwest slopes. Are they really acuter? We're not sure yet. But that's not highly relevant to the Hunter. What is relevant is that we managed to genotype 16 stands from in the catchment, which basically represents all the remaining stands. So you can see how, you know, once upon a time it would have been all along the Hunter River here, but we're really reduced to just these few stands. As I already said, they're all genetically subspecies camaldulensis or hybrids with other red gum species. And we also found that they're most closely genetically related to stands in the Namoi River, with Namoi River catchment, which is actually this one to the northwest here. So it looks like the catchment's actually been colonised from the Namoi River catchment. If you ask any Hunter local, they're going to look at you funny because there is a Liverpool range which sits right on the border between the two catchments there. So how it's managed to cross that range, we don't know, but that's what the genetic patterns are telling us. We also find, probably related to that same thing, that it's a recently colonised catchment, that we see lower genetic diversity on average in stands in the Hunter than what we see in the rest of the Murray-Darling Basin. So basically the story here is that probably somewhere in the recent past it's managed to have a single colonization event into the Hunter from the Namoy River catchment, which is interesting and does have implications for how we then manage and conserve these stands. All right, aim 4, looking at patterns of remaining genetic diversity in the Hunter. So these two maps here, the various stands are coloured by how genetically diverse they are. The more blue, the more diverse they are. The more red, the less genetically diverse they are. So to calculate these parameters, we need a minimum number of samples. So a few of the stands dropped out here and removed all the hybrids, obviously, because that can affect it. But some general patterns we can observe here is that genetic diversity is highest up near Aberdeen and some of these other sites down here. These are all sites on the main channel of the Hunter River. Once you start moving to some of these tributaries where you get populations on the Goulburn River here or Wallumbay Brook, you see lower genetic diversity. So, basically we think, it's colonised via the main river channel, and that's where the diversity is highest. You get, you also see higher rates of hybridization in these tributaries. So, core habitat is the main river channel, basically, which, is where the flooding events that support the species would have occurred. Also is maybe a pattern of sort of higher genetic diversity upstream, decreasing as you move downstream, but that was a bit less clear. And that might be due to the land clearing being more intense downstream. All right, so then moving on and looking at the seed collections by the Landcare group. So as I said, they got 10 maternal lines. They were tracked the seed in those maternal lines. They tracked that into a nursery. We were then sent leaves from the seedlings in the nursery. Differing number of seedlings per line, but all of them were more, relatively evenly represented. Those 10 maternal lines came from 4 stands along Wallumbay Brook, which is the area Bulkbroger Landcare works in. 3 of them came from very small. One of them was a lone tree at which end is which? I think Broke end is the Bulger end is downstream. I don't know. Which is upstream, really small stands, and then 7 of them come from the stand at, I think Bulger is the downstream. I'm confused there. But yeah, basically we see that the stand where we get the 7 maternal lines, which was a bigger stand where there's more remnant trees, has more genetic diversity captured in those seed lines than the ones where we only have, say, 1 tree left or a couple of trees left. So that's here, the green, the higher values, higher genetic diversity, basically. We also see fewer inbred seedlings than smaller stands, which is very good because we know in Eucalyptus, that they do inbreed quite highly. So people will say they got an outcrossing. That's all post-zygotic. So they'll produce a lot of inbred seed. Natural systems up to like 50% of the seed produced is actually selfed. But then those seedlings perform really poorly once they germinate, or they germinate really poorly and then they have really low competitive advantage, and you end up with an adult population which is pretty much 100% outcross seed. So what we can get here is if we collect from these small stands, we get a lot of selfed seed because there's no other pollen available, basically. But then those seed lines will perform very poorly when you plant them out because they're all inbred. And because we track these lines, we're able to go to the nursery folk who had germinated these in the nursery, and they are able to confirm, yes, these seed lines performed poorer, they germinated more slowly, grew less vigorously, which corresponded well to our expectations. We also, I mentioned earlier, the F1 hybrids between Amphifolia and camaldulensis along Wallumbay Brook. One of the parent trees we were able to determine was an F1 hybrid. So we took that back to the Landcare group. They said, okay, well, we don't want to plant out that seed if it's all going to be of hybrid origin. They were able to then remove that maternal line from their, they didn't even plant it out. They didn't need to remove it. It never got planted out. So that's, I guess, evidence for making sure you track your maternal lines, your seed lines. You can then go remove anything that's bad, increase stuff that's good. Yeah, so that's all I say on that. And then the final aim was sort of optimizing seed collecting strategies to establish healthy restored populations. Part of that is obviously climate change adaptation. Although maybe that's less relevant in the Hunter because there is a really strong climate gradient in the Hunter already. So in green here is basically the area of occurrence of camaldulensis currently in the Hunter. If we then climate match for mean annual precipitation, mean annual temperature, we get this blue area, which is very widespread in New South Wales under current conditions. When we then project forward what climate is projected to be in Hunter in 2070, then match current conditions to that, we get this red area, and we find actually these populations are probably going to be all right because the climate isn't going to be outside their realised envelope. But we can also go to these catchments to the northwest and get extra seed material that might be more genetically diverse and pre-adapted to future climates, and include that in our restorations to help buffer them against the effects of climate change. Which is good because that fits with the genetic findings that the Namoi River to the northwest here is where it is most genetically similar, so we can go get genetically similar but more diverse and climate-ready seed from the Namoi River catchment. And then we did a number of optimizations using a very similar approach to what Saphira did for different restoration sites or scenarios. So the first one here is just how many maternal lines do we need to collect in the Hunter Valley to capture 90% of common alleles. We found that you need to go to 4 sites, collect 5 maternal lines from each site. So if you have 20 seed lines total, you've captured pretty much all the genetic diversity present in the Hunter. The next 2 are not particularly analytical, they're just scenarios that we can posit and say, okay, if you're concerned about climate change, perhaps you want to bring in 1 external site. What you want to do is make sure you capture your local genetic diversity as well, and then in addition to that, add 1 site, or if you want to go, say, 50% climate ready, capture your local genetic diversity and bring in an equal number of climate ready provenances. We weren't going to give any recommendations on that because we also don't want to homogenize all the populations, and so we don't want to say, okay, go to this stand here and then everybody goes there and everybody's using the same seed. We want to say, okay, we'll go to that catchment and hopefully people go to different stands in that catchment and we get more diversity in there. And then there were a couple of scenarios here just supplementing existing sort of populations in the Hunter. So Muswellbrook City Council is interested in supplementing populations they've got in the middle of Muswellbrook, parkland there, which is actually quite genetically depauperate, and some of it looks like it's planted from elsewhere in the Hunter in the first place. So we were able to say to them, okay, well, you have, this is the genetic diversity in your stand already. What sites do you want to go to that most complements your existing genetic diversity? Write the recommendation down back to them. And then also for the land care group, say, okay, here's the genetic diversity of those maternal lines you've already got. How then do we add more genetic diversity because you've collected them all on Wallumbay Brook where it's all not great. So yeah, just in conclusions, we were able to show that hybridization between Eucalyptus camaldulensis and other red gum species is frequent in New South Wales, including in the Hunter. And I didn't touch on this, but basically those hybrids tend to not be on main river channels, so when you get into tributaries like we see in the Hunter. So that means if you want to restore river red gum, go get your seeds from a stand that's on a large river or a large river plain. All stands of the Hunter are subspecies camaldulensis, and where the morphology differs, it's probably due to hybridization, not because of the different subspecies. The Hunter catchment population is part of the wider Darling catchment lineage and is likely the result of a colonisation event from the Namoi River catchment. Genetic diversity is highest in larger stands along the main Hunter River in the Hunter catchment, and once you get onto the tributaries, you sort of see lower genetic diversity. All populations on Wallumbay Brook have experienced introgression from Eucalyptus amplifolia, which means that the seed collected along Wallumbay Brook by the Lanka group is fine to use on Wallumbay Brook because that's the natural patterns, but you then don't want to be taking that elsewhere because that's pretty much the only place you'll find those hybrids naturally in the wild because it's the only place those 2 species co-occur. And we were able to develop specific recommendation scenarios using this genetic data to address a number of needs for different stakeholders. And so I just want to finish by saying, as I said at the start, this is work I did while down in Sydney. I'm now based at the Queensland Herbarium. We're looking to set up a conservation genetics program up here. So, if you are from Queensland and the last 4 talks have inspired you to look into some conservation genetics, do come and talk to me. It'd be great to have a chat.